this is chapter 12, which is all about nervous tissue and the neural impulse essay. So it has some really important things in it that need to be covered. So let's just kind of start at the beginning. I did fill in a lot of words on the slides, so I don't have to write as much. So we're going to talk about the cells of the nervous system. So what kind of cells make it up, how the neuron actually fires, what a synapse is, and we'll talk a little bit about how neurons communicate with each other. So this is actually a picture of a synapse. So synapse is the intersection or the junction between the end of one neuron and the beginning of another neuron or a muscle or a gland. Okay, so the general functions of the nervous system I have listed out. So they're going to collect information. So we're going to take information from the outside, usually from the environment or from something internal that's going on. And what they're going to do is receptors are going to collect that information, and they're going to monitor what's going on around you and communicate that to your central nervous system so your body can decide what to do. Okay, so collect information. So what's going on around you or what's going on inside of you. Okay, then they're going to process and evaluate the information. So they're going to take that in, the information in, and they're going to say, okay, well, this is light information. She needs to dilate her pupils so that she can get more light in so she can actually see because it's dark. Okay, so they're going to process the information, they're going to evaluate it, and then they're going to initiate a response to the information. Okay, now the effectors, or the things that are going to be able to respond to the nervous system, are all three types of muscle, glands, and other neurons. Okay, so those are things that are going to make a response occur. Okay, now anatomy, right? We're always subdividing everything and renaming everything. So they have it organized a couple different ways. So one thing is the structural way that things are subdivided. And this picture doesn't look too pretty when I imported it into this GoodNotes software, but you can see, okay, so the central nervous system is one of the structural subdivisions, and it's the brain and the spinal cord. So it's what's in this peachy color here. So the brain and the spinal column, the spinal cord, or the CNS is how we refer to it. Okay, the peripheral nervous system is all the stuff that comes out from there. Okay, so the peripheral nervous system is our nerves and ganglia, and we'll talk about what ganglia are here in a minute. And it's what is in this yellow color in this picture. Okay, so central and peripheral. So that's like anatomical differences. So brain and spinal cord is the CNS, and then the PNS is everything outside of there. Okay, now here's some vocabulary. So a nerve is a bundle of nerve fibers. So you could pretend that you're taking a whole bunch of spaghetti and you're taping them together. Okay, so each little spaghetti noodle is a nerve fiber, and if you tape them together, that's going to make a nerve. Okay, now the ganglion, so ganglion is singular, so it's like a ganglion, so ganglion, and then plural is ganglia. Okay, those are the cell bodies of the neurons, so that's where the organelles are going to be. I'll show you in a picture here in a second what these actually are, but we just want to get those two definitions in there so you know what they are. Okay, now, functional organization. So how do we organize it functionally? Okay, so how do they actually work? So we have two categories here. So sensory is information that's going to come towards the central nervous system. So another word that they use for sensory is afferent. Okay, afferent with an A. Okay, so that means we're going to carry information from a receptor towards the central nervous system. So I always think of like if you touch a hot pan, Okay, the pan information, that the information that the pan is hot is going to come from a receptor in your finger, and it's going to travel to your spinal cord and your brain, and that's sensory information. Okay, now the information that says what you're going to do with that finger, whether you need to pull it away, is motor or efferent. So that means it's exiting the central nervous system. Okay, so it's coming away, and I'll show you pictures of it again. Okay, so I always think efferent is exiting, and it's E and E. Okay, and afferent is A, and it comes before the E. So afferent means it's coming towards the central nervous system, and efferent, it's exiting. Okay, now another way that we categorize it, okay, is somatic and visceral. Okay, so somatic is body. Okay, so it's the body system. Okay, so somatic stuff is what's in your body, okay, and that what we can be conscious of. Okay, so muscles, joints, bones. Okay, so the receptors for the somatic nervous system are the eyes, the nose, the tongue, the skin. So touch, taste, smell, vision. Okay, so somatic is another way we break it down. And then visceral, okay, is within your body. Okay, so you don't think about it. So the organs in your thoracic and the abdominal cavities, and also things within your blood vessels. So visceral can also be considered autonomic, so you're not thinking about it. Okay, so you're not thinking about what's going on in your heart. You're not thinking about, is your food moving from your stomach to your intestines? You're not thinking about whether your blood vessels are going to dilate or not. Okay, so somatic, we're actually conscious of it. And then visceral, it's more automatic. We're not thinking about it. Okay, now, well, I think we're okay. Okay, sorry, I'm just trying to make sure that I get everything that you need. So I'm looking at my notes that I have written down. So a visceral and the somatic system are going to have sensory and motor function. Okay, so sensory and motor will be part of this and part of this. Okay, but for right now, I just want you to understand the definitions. Okay, now here's a picture that tries to explain what I was, or explains better what I was trying to tell you without a picture. Okay, so here's a finger. So say you picked up a piece of broken glass and you stuck it. Okay, so sensory or afferent neurons are going to take information from the receptors in the finger. Okay, and they're going to take it to the central nervous system. Okay, so out here, okay, everything in this green box, that's peripheral, it's PNS. Then as we get going, right, we'll get it to the central nervous system. There are interneurons, which that's a new term we haven't talked about. So interneurons are also called association neurons, and those are neurons that are within the CNS. Okay, so they're going to be within there. They don't leave it. So there might be a connection in here, kind of like they're showing. Okay, so here's a neuron. Okay, then it goes to the central nervous system. There might have to be a relay of information there. Okay, and then information is going to come out, and we're going to have motor or efferent neurons, and they're going to take the information to the muscle in your finger and your arm, and it says, move your finger away from that glass because you're going to cut yourself. You just cut yourself. Okay, so sensory, afferent, or afferent, right? The right way is afferent. Okay, and then there's interneurons, and then there's motor. So sensory is going to come in, and motor is going to come out. So see how the arrows are going. That's important. Okay, so sensory information is going to come in. It's going to go to the nervous system, central nervous system, right? And then it's going to come back out and cause a change to happen. Okay, now they have it all organized in the book to show you how it's all laid out. So this is a nice way to see it. Okay, so sensory is going to detect the stimulus and it's going to take the information to the CNS. So it's going to the brain and the spinal cord. Okay, now there's two parts of it. Okay, so there can be somatic sensory. Okay, so that means it's coming from the body. Okay, and you're consciously perceiving it. So that means you're aware of it. Okay, then there's visceral. So visceral is coming from the internal organs. Okay, you're not aware. You're not thinking about it. You don't really have any control over it. Okay, now over on this side, we have motor. Okay, so motor is going to go from the CNS to the effectors. Okay, so it's leaving the brain and the spinal cord. I hope this is recording. I'm going to double check here, guys. Give me a second. Oh, come on. Yep, it's still recording. Okay, just double checking. Okay, so somatic then is voluntary. Okay, so you're consciously aware of it. So skeletal muscle. So remember, skeletal muscles are what's attached to your bone. Okay, the autonomic output is you're not conscious of, so it's involuntary. So it's things that you don't have control over. So involuntary things are going to be the heart muscle, smooth muscle, and glands. Okay, so your heart, the smooth muscle in your gut, okay, or your blood vessels, and the endocrine glands or salivary glands. Okay, so the information is coming in here, right, and it's exiting over here. Okay, now let's talk about neurons. Okay, so neurons, the fundamental or general characteristics of a neuron are these 1, 2, 3, 4, 5 things, right? So one is that they are excitable. Okay, so they can respond to external stimulus in the environment. So they know what's going on. Okay, they can initiate a response. Okay, depending on where they are, determines how they're stimulated. Okay, so if it's in your eye, it's going to be stimulated by light. If it's in your ear, it might be stimulated by sound. Okay, neurons are also conductive, and we'll talk about that in great detail. Okay, so they spread an electrical impulse. So they're going to take information and they're going to move it along. They can secrete. So they release these chemicals called neurotransmitters, and the neurotransmitters are going to be able to communicate with they're going to be able to communicate with muscle, glands, or other neurons. Okay, they last a really long time. Okay, so they last your lifetime unless you've had some sort of significant damage. Okay, they do lots of research, like if you could fix a spinal cord or if you could fix nerve damage, it would be totally amazing. It would change somebody's life. Most of the time, they don't divide after fetal development, but they're amitotic. Okay, so they don't go through mitosis after the fetal development. There's a lot of research going on to try to figure out how to make them reproduce, right? And how to keep them, not keep them alive, but if you could get them to copy themselves, if you could get them to go through mitosis, you could heal some major accidents. Okay, sorry, I'm babbling. I hope this makes sense. Okay, now here's the structure of a neuron. So we're going to go over what the parts are of this. And there's a lot of parts. Okay, so let's see. Let me start down here. Okay, so down here at the bottom, okay, these are called synaptic. I call them synaptic bulbs. I'm looking at the new edition of the book here. Let's make sure. They're calling them synaptic knobs. This is one of the things they kind of maybe change what they actually call it. So all of these little things are where a synapse is going to form. Okay, so synaptic bulb or synaptic knob. Okay, this is the soma, or we also call that the cell body. Okay, so this is where the organelles are going to be. So in here, right, there's a nucleus, there's a nucleolus. And this picture in the 10th edition, it's on page 444. Okay, so I'll put it down here. It's a little bit different. It's pointing a different direction, but it's the same idea. Okay, now these little things that are pointing out that look kind of like tree branches are called dendrites. Okay, dendrology is the study of trees. So dendrites are tree-like branches that are coming off here. So these are dendrites. Let me write it down here. These are cytoplasmic extensions. Okay, so they're going to receive a stimulus. Okay, so from light or whatever external environment. So this is where the signal is going to come in, or at these dendrites and these branches. Okay, now the cell body, it has no centrioles, right? Because no mitosis. It has the nucleus, it has the nucleolus, it has mitochondria. It has lysosomes, Golgi complex. It does have something different. It has these things, they're called Nissl bodies. And Nissl bodies are clumps of rough ER. So if you think about rough ER, those have ribosomes. And what's the purpose of a ribosome? Okay, so the ribosomes are the site of protein synthesis. So these guys are going to make lots of proteins, which would be neurotransmitters. Okay, they have these things, they call them neurofibrils, which those are part of the cytoskeleton. I think I got most of it on here. Okay, now this part of this whole thing is called the axon hillock. Okay. So it's kind of like its neck. So it's like a little hill. Okay, and then this is the axon. Okay, so the axon is the long part. Okay, this axon is actually wrapped in myelin. And we're going to talk about myelin here in a minute. Okay, so it's got myelin. It has these spaces in between the myelin. Okay, so in here, let me try to, if I change colors. Okay, so spaces in between the myelin, and those are called, oh, they're calling them something different. They're calling them a neurofibril node. We used to call them nodes of Ranvier. We're trying not to name things after people anymore, so they're called a neurofibril node. Okay, so those are the spaces in between the myelin. Let me see. What else do I have? I think that's it. That's everything on there. So there's lots of parts, right? Let's highlight it and we'll kind of go through some of them again. So, okay, soma is the cell body, right? The dendrites are these extensions that are coming off the ends. They look like tree branches. The axon hillock is kind of like the little neck of it. This is the axon, so it's the long part, and that's what's going to be the extension to the next piece. Okay, synaptic knobs are at the ends. Okay, myelin is what's going to wrap around the axon. These neurofibril nodes are the spaces in between the myelin. Okay, and then all of this stuff up here, these are all things that are in the cell body. Okay, so these are just what's inside the cell and what makes it function. Does that make sense? So this is one big cell, and it's got all these different parts. All right, let's go on. Okay, now, this, I don't get too hung up about this. I just want you guys to recognize that this exists. Okay, I show you this picture. This is like when we draw a neuron, this is what we think of, okay? And it's shaped like a star, right? It's like an astrocyte. It's shaped kind of like a star. I think it looks more like an alien personally, but kind of like a star. Okay, just so you know, they're not all shaped the same way, right? They can be organized in different ways. So they can be some that have one axon and a whole bunch of dendrites. Okay, so this is the most common, and that's called a multipolar neuron. Okay, so that's most everything in your brain, and your spinal cord is going to look kind of like this. Okay, it's not simple. It's got a lot of branches, so there's lots of information coming on. And okay, there are bipolar neurons. Okay, so they have one axon and one dendrite. So they're much more simple. There's unipolar. Okay, so if you look at cell body, it's kind of off to the side. And then an anaxonic neuron, so it doesn't have an axon. Okay, I just want you to be familiar. No, multipolar is the most common, right? Bipolar, we'll talk about when in the nose and the eye and the ear. Okay. All right, now there's two kinds of transport in a neuron. Okay, so you got this big neuron, right? And let me draw a picture. Okay, so if we go here, right, so here's my cell body, right? And then there's my synaptic knob, and there's the nucleus. Okay, so up here, okay, this is where we're making proteins. So these proteins that are being made up here have to be transported down here and down here. Okay, so what happens, they call that axonal transport. Okay, so some things move this way and some things will move this way. Okay, so anterograde transport, okay, it's going to move down the axon and away from the cell body. Okay, so this is anterograde right here. Okay, retrograde, so if you think about retro, right? Retro's backwards. So retrograde transport, I'm going to make it blue to match what I underlined it with. It's going to go the other direction. So just that we make lots of compounds up here, but they're going to travel back and forth. Okay, now a nerve and a ganglion. Okay, so the structure of a nerve and a ganglion. And do I have, yeah, I have it written out over here. Okay, so I kind of like to pretend with this. I try to imagine, for some reason, this is always hard for me to imagine. So I want you to kind of think of if you had spaghetti noodles and you wrap the spaghetti noodles up, okay, like taped them up with one color tape, and then you put them in a bundle, and then you tape that up, and then you tape the bundles together. That's kind of how a nerve works, okay? So if we look here, okay, here's the axon, Okay, so this is what I just drew in that picture. So this is an axon. And what we'll do is the axon is actually covered in this thing called the endo. So endo is inside, right? And neurium. Neurium is the wrapper. Okay, so the axon and the neurolemnocyte, okay, are wrapped in the endoneurium. Okay, now if we take a whole bunch of those and bundle them up, okay, we end up with a fascicle, okay, or a bundle, okay, and they're wrapped in what we call the perineurium. Then we take a whole bunch of those and we put them together, and that's when we get a nerve, okay, and that's wrapped in what's called the epi. So epi is on the outside, so the epineurium. Okay, so maybe it's easier to see in this picture. Okay, so if we look here at this black and white picture, let me try to find a color, a bright color. Okay, so if this is an axon and the neurolemnocyte, right, and those are wrapped up in the endoneurium, and there's a whole bunch of them in here. Okay, and then let's say we take the pink, right, and we go out here, right, and that's the perineurium. Okay, so we're wrapping a whole bunch of those up together. Okay, and the perineurium is the outer wrapper. Okay, the fascicle is the bundle. Okay, and then we take those bundles and we wrap them up even more. Okay, and when we wrap those up even more, then that's when we get a nerve. Okay, so the nerves you could actually see, like if you could see a nerve, maybe like if you were dissecting a chicken breast or you're cutting chicken up, you could see a nerve in there. Okay, so nerves are bundles. And now think about like if you are trying to think about how to think about how wires are made, right? Wires are made in bundles. So that's kind of how our nerves are made. Okay, so epineurium. So I have dense, irregular outer tissue. Perineurium. Okay, this wraps the fascicles. Endoneurium wraps the individual. And then I have this is kind of random in here, a ganglion. Okay, we'll have to talk about that. Okay, so this is from the outside to the inside. Okay, so epi, peri, endo. Okay, now a synapse. I need to, where do I have ganglion? I'll get to ganglion, hold on. Okay, so now a synapse. A synapse is where two neurons or a neuron and a muscle or a neuron and a gland are going to come together. Okay, so in this picture here, let me try, I want to find something that you guys can actually see that's bright. I don't know, let's try this. Okay, so in this picture up here, okay, there are two neurons together. Okay, so this is the presynaptic neuron, and this is the postsynaptic neuron. Okay, so this neuron is going this way, right? Here's the synaptic knobs, and those synaptic knobs are communicating with the dendrites in the postsynaptic neuron. Okay, now, the pre and post, it's not an assigned name, all right? It just means that there's one before the other one. Okay, so if you look at this, okay, here's the knob, okay, or the knob, the synaptic knob. Okay, and then here's the postsynaptic neuron dendrite. Okay, or it could be the cell body. Okay, now what's going to happen is information is going to come down that neuron. Okay, and then there are vesicles in here. The vesicles have neurotransmitters in them. Okay, so the neurotransmitters will be released, which we'll talk about. They're going to cross this space between the knob and the next neuron. Okay, they don't touch. There's space in there, and that space is called the synaptic cleft. Okay, so it's got to cross the cleft, and then it'll bind to receptors, which are proteins on the membrane, in the membrane of the next neuron. Okay, now we want to talk about what neuroglial cells actually are. Okay, so glial is like glue. Okay, so nerve glue is how I always think of this. Oh shoot, don't allow. What happened? Cancel. Okay, I clicked on the camera button instead of the pen. Hold on. No. Nerve glue. Okay, so neuroglial cells are cells in the nervous system besides the neurons. Okay, so the way I kind of think of this is the neurons are kind of like the rock star. Okay, and the neuroglial cells are the entourage that supports the rock star. Okay, so there are only so many neurons and they don't go through mitosis. Okay, so they can't regenerate. Now, so they need support, they need help. All right, so they're kind of like the divas. So they're going to need support from the other cells in the nervous system. Okay, so this is a picture of central nervous system. So central nervous system would be brain and spinal cord. So if we look, okay, here's a neuron, right? Here's a neuron. Now there's all these supporting cells around them. So their entourage is there to make sure that they have what they need. Okay, so on this picture, so microglia, okay, see these little cells. We have astrocytes, which kind of shaped like stars. We have ependymal cells, and then we have oligodendrocytes. Okay, so I'm going to explain these and we can come back to this. Okay, now neuroglial cells, there's 6 types. 4 are in the central nervous system and 2 are in the peripheral nervous system. Okay, oligodendrocytes, I always kind of think of them, they're kind of like an octopus. And what they do is they're going to produce the myelin sheath. Okay, so if we go over here, here is a oligodendrocyte and it's making this myelin sheath. Okay, I haven't talked about the myelin sheath yet, and I can't remember where I have it in the notes, so let's just talk about it. Okay, so the myelin sheath is made out of lipids and it wraps around the axon. Okay, so it wraps around the axon. What they do is they speed up, causes the neural impulse that's traveling down this to speed up. Okay, and we'll talk about how that happens here in a couple minutes. But myelin, it's there, it makes your neural impulse travel faster. So the axons that are wrapped in myelin can get information to a part of the body faster than if there's no myelin. Okay, now MS, multiple sclerosis, is a disease of the myelin sheath, where the myelin sheath gets damaged. So then the information doesn't travel as quickly. Okay, now astrocytes are another type of cell. They're shaped like stars. They are the most abundant neuroglial cell. Okay, now what they do is they're gonna pretty much take care of the neurons. Okay, so they're gonna nurture, support, protect, and guide the neurons. So they're gonna regulate the fluid around the cells in the brain. They're gonna be part of the blood-brain barrier. They're gonna provide structural support, and they're making sure, they're like nannies maybe, or parents. They're making sure that the neurons have what they need to survive. Okay, so astrocytes are the green ones on here. Okay, ependymal cells. If you look at these, what do they remind you of? They kind of remind you of epithelial cells. Okay, so ependymal cells are going to line cavities in the brain and in the spinal cord, and they produce this fluid called cerebral spinal fluid. Okay, now the other type of cell are microglias, and they are part of your immune system. And they are actually macrophages. So macrophages are cells that can do endocytosis, right? So they're going to gobble up things. So where are they at on here? Microglia are little, okay, micro, right? So micro and macro you could put together. So they're going to go through, and if there's something in here that looks like it doesn't belong, they're going to engulf it and destroy it so it doesn't affect the rest of your brain. Okay, now, neuroglial cells can go through mitosis. Okay, axons can't, or not axons, neurons, right? No mitosis. So if someone has a brain tumor, okay, if your neurons can't go through mitosis, they can't develop cancer. Okay, they don't have the stuff to do it. That's not what they're there for. Okay, but these guys can. Okay, so if you have a brain cancer, it's one of these kinds of cells. Okay, now in the peripheral nervous system, we have two more types of these cells, and they kind of work the same way as two of the other types. Okay, so we have satellite cells, and they're in the ganglia, and I haven't explained ganglia yet, of the peripheral nervous system. Okay, and they're going to regulate the chemical environment. They're going to take care of the neurons in the peripheral nervous system. And then we have neurolemnocytes, which we used to call Schwann cells. And these are myelinate. So these are like oligodendrocytes. They're not the same. So oligodendrocytes are in the central nervous system. Neurolemnocytes are in the peripheral nervous system. But they both make myelin. Okay, so they both make the impulse travel faster. Okay, here's where the myelin sheath is. Okay, so the myelin sheath is the insulating layer around the nerve fiber. And I have a picture here. Okay, so here's the axon. Okay, axoplasma is just the cytoplasm. Axolemma is just the membrane around it. Okay, and then what it is, it's wrapped around it. So if you took like a tube of toothpaste and rolled it up, but you had like a pencil in the end of it, or you took a piece of paper and you rolled it up. So if you had something like this and you just rolled the myelin around it, that's how the myelin sheath is set up. Okay, they're 20% protein and 80% lipids. Okay, so when kids are younger than 2 years old, right, they drink full fat milk or whole milk. That's a better word for it. Okay, the point of having lots of fat is because they need the fat to make the myelin, so for brain development. Okay, now the spaces between where the myelin is, so here's myelin, here's myelin, here's myelin. The spaces between are called the neurofibril nodes. We used to call them nodes of Ranvier. Okay, the electrical impulse, and it'll make sense when we talk about it, it actually jumps, and it's kind of like a video game with a booster. So the neural impulse with these myelin sheaths, it'll kind of hop through where the myelin is wrapped. Okay, so they get like a little turbo boost or power surge, and they can move faster. Okay, now there is initial segment on the neuron that's between the first Schwann cell and the axon hillock. Okay, and this is called the trigger zone. So that's where an impulse is going to begin. And then here's what I was just trying to tell you about the whole milk. So myelination begins at the 14th week of gestation. Okay, and it occurs during infancy, so probably till about 2. Okay, so there's a picture of myelin again, and it's a cell that's doing it, that's wrapping it. So it's either oligodendrocytes in the CNS or the neurolemnocytes in the PNS. Okay, and really, really, really important for your nervous system to function properly. And there's another picture that I have of the same thing. Okay, so myelination by oligodendrocytes. Okay, so one cell might myelinate a whole bunch of axons in the CNS, which is right there, right? But in the PNS, it's more like 1-to-1. So you have one axon that's long, and it's got a whole bunch of different cells on it. Oh, so then here I got ahead of myself. Okay, so 2 disorders that affect the myelin. Okay, one is MS, so multiple sclerosis. So what happens is there's demyelination. So it's an autoimmune disease. So something happens and your body starts to attack the myelin in the central nervous system. There are a whole bunch of different types of MS, but basically what it's going to do is it's going to impair the sensory information coming in and the motor information going out. Okay, another thing is, I always butcher this, Guillain-Barré syndrome. Okay, so you hear people pronounce it different ways. So with it, the inflammation, it's in the peripheral nervous system, okay, and the roots of the spinal nerve. So your muscles get weak. People might have flu-like symptoms. They believe Zika could have long-term effects that actually cause this, so Zika virus. Okay, now you don't have to memorize these numbers. Okay, I just want you guys to get the idea that the speed depends on how big the fiber is, the diameter, and whether it has myelin or not. So the big thing is that you get the idea that things are traveling, information is traveling super fast, right? So it's traveling 1/2 to 2 meters a second in the slowest ones. Okay, but in the large myelinated fibers, it's going 120 meters a second. So the information is getting sent before you can even think about it or realize that it's happening. Okay, now peripheral nerve fibers have been shown that they can regenerate. Okay, so if the soma or the cell body is okay and some of the neurolemma is present, then it's been proven that they can regenerate. It just takes a while. Okay, and here it goes step by step how they can regenerate. So if there's trauma in the middle, right, but there's some of the neurolemma left, okay, it can get sealed off and then it can regenerate. Okay, if the central nervous system is damaged, it can't regenerate. Okay, so that's why, right, we don't want brain damage, we don't want spinal cord damage. Okay, now, how does this actually work? So what's going to happen? Okay, so it's all based on electric potential. Okay, so we're going to talk about ions and cations and anions, okay, and currents. So one big thing to remember is that the nervous system, it's a bunch of cells connected together. it's not one direct line going from like your toe to your brain. Information has to go from one cell to another cell. Okay, the neuron communication is based on electrical potential. And living cells, okay, so if cells are alive, they're actually polarized, so they're charged. Okay, so they're going to be charged. Okay, now I have some videos that explain how a neural impulse fires. So I have two of them that go together. I'm trying to think. I'll post those two. So it doesn't have all the other stuff in it. It just has this part of it. Okay, so you have to remember pumps and channels. So these are things that are located in the plasma membrane. Okay, so pumps are things that are going to go against the concentration gradient. Right? So we're going against it. So it's going to be active transport. Okay, we have things that are called leak channels. So leak channels are always open. Okay, and things are going to move down the concentration gradient in them. Okay, so things are going to just kind of leak through. Okay, we have chemically gated channels. So these I usually say C, J, C. Sometimes they call them ligand gated channels. Okay, a ligand and a chemical are the same thing. So basically what happens with these guys is you have to have a chemical that's going to stimulate the protein channel to open. Okay, and then we have voltage gated channels. Okay, so voltage gated channels are going to open when they are electrically stimulated to open. And I call these VJCs, electrically stimulated to open. Okay, so we're going to have a change in charge. So you're looking for a change in charge, right? Right now, I don't know whether to go ahead and do this on here or make you guys go to the other video. I'll just put it on here and then it'll all be in the same place. Okay, so here's a neuron. Okay, and my neurons end up looking like Bart Simpson. Okay, so there's the soma, the cell body. There's the axon hillock. Here's the axon. And there's the synaptic bulb. Okay, now I'm wondering if I can copy this for later. What do you think? Think I can do it? I'm not sure I'm smart enough to do it. Got it. Oh, wait, I don't know. Copy. Okay. All right, now I just want to delete that. Okay, so let's go back here. Right, so, right, you know the parts. So these are supposed to be dendrites. Right, cell body, axon hillock, axon, and then synaptic node. Okay, now, part of figuring out how an axon works and how a neuron fires has to do with where the pumps and the channels are. Okay, so what we're going to do is we're going to label this with the pumps and the channels. Okay, so the first thing that we're going to do is we're going to do the sodium potassium pumps. Okay, so sodium potassium pumps, I'm going to make blue. Okay, now those are Sodium potassium pumps are, I'm looking for my notes because I have all these, I don't want to mess you guys up. Although I've only done it like a gazillion times, right? Sodium potassium pumps are going to be, they're going to be all over, okay? So they're all over the cell body, all over the dendrites. They're everywhere. Okay, they're the entire length of the neuron. I'm just going to write that here instead of trying to draw them here. Okay, and I'll try to explain what more about them here in a minute. Okay, so sodium potassium pumps are the entire length. The other thing that's the entire length are sodium and potassium leak channels. So they're all over. Okay, they're everywhere. Okay, now the way that sodium potassium pump works, let's see if I can try to put I want to just add a page. Let's see if I can add a page right there. Can I add a page? Will it keep going? Yep. Okay, so the way that these work, okay, so if I draw it like this. Sorry guys, I hope this is okay. Okay, so sodium potassium pumps, for right now I'm going to draw the pump down here. And then the leak channels up here. Okay, now, what's going to happen? Okay, so let me change colors here. Okay, so here's the dendrites, right? And here's the cell body. Here's the sodium potassium pump. And then these are leak channels. Okay, the sodium potassium pump, what's going to happen is it's going to pump 3 sodium out for every 2 potassium it pumps in. Okay, remember it requires ATP. Okay, so 3 sodium out for every 2 potassium that go in. Okay, now I want you to think about that. Okay, so we're getting 3 out, 2 in. So that means that the inside, okay, is going to end up being negatively charged. Okay, so it ends up being negatively charged. Okay, at the same time that the sodium potassium pumps are working, and this is happening all the time, right? Most of the energy that you make goes to run these sodium potassium pumps. Okay, at the same time, what's happening, okay, is that we're getting excess, what color do I have here? Blue. We're getting excess potassium inside, right? Because it's getting pumped in over and over and over again, right? And we're getting excess sodium outside because it's getting pumped out over and over and over again. Okay, so we're starting to lose equilibrium. We're not consistent. So what has to happen is that the potassium will start to leak out because they're at a higher concentration out here. Okay, and the sodium will start to leak in. Okay, but the resting membrane potential, which I have on the next slide, becomes established because of these sodium potassium pumps and these leak channels. Okay, overall, the inside of the cell is negatively charged, and the magic number is negative 70 millivolts. Okay, so sodium potassium pump is working opposite, right? It's active transport. So it's pumping 3 sodium out and 2 potassium in constantly. Okay, that gives us a negative 70 charge inside the cell. In order to not run out of sodium and potassium, what's going to happen is we have these leak channels. So as the potassium builds up inside the cell, it'll leak out to the outside of the cell, and the sodium will start to leak in. So those are passive. They're going to move down their concentration gradient. Okay, so that's the first part. Right, now let's go here. Okay, so they're everywhere. Let me try to, I think I'm gonna, this is messy and I don't like the way it looks. Let me try to find the eraser. Hold on. Where's the eraser? There's the eraser. I don't know if I can, well, I'll just erase all this. Okay, let me go back and try to paste. I wonder if it'll let me just paste. Not smart enough to figure out how to paste. All right, so here I'll draw another thing. Ah, no, off. Okay, here we go. Okay, now other channels that are going to be on here. Okay, so we're going to have voltage gated channels and ligand gated channels. So out here, okay, we're going to have these orange ones. We're going to have chloride ligand gated channels. We're going to have potassium ligand gated channels, and we're going to have sodium ligand gated channels. Okay, and those are going to be on the dendrites and the cell body. Okay, so they're going to open when a chemical binds to them. Okay, the voltage gated channels that we have are going to be down here. Okay, the voltage gated channels are on the axon hillock and the axon. And there's some down here too. Okay, so they're going to open when the charge changes. Okay, so let's just go forward for now. Okay, so the difference between the extracellular fluid and the intracellular fluid is negative 70. Okay, and that is established by these sodium potassium pumps in the leak channel. The membrane's really permeable to potassium, okay, but big anions can't leave, and the membrane's less permeable to the sodium. So basically what happens is you end up with the inside of the cell equaling negative 70. Okay, so that's resting. Okay, so here is a picture of what I was trying to draw for you. Okay, so here is what's inside the cell. Okay, here's what's outside the cell. Okay, and if you could measure the charge of it, okay, you would find that the inside is more negative than the outside, basically. Okay, and that's all you need to know. That's good. Okay, now we can change the resting membrane potential. Okay, so we say it's polarized. So basically, two things can happen to it. We can depolarize it, or we can hyperpolarize it. So when we depolarize it, what we're going to do is we're going to change the signal from negative 70 to something more positive. Okay, so it's going to go up. When we hyperpolarize it, it's going to become lower. Okay, now, graded potentials versus action potentials. So things can occur two different ways. So an action potential is all or nothing. Okay, so it's on or it's off, it's going or it's not going. Whereas graded potentials are more like a volume control. So they can be loud or soft. Okay, here it just explains the difference, okay? So a graded potential, here's how things are changing. So they're chemically gated, the voltage change is going to be really small. It depends on the stimulus, whereas what we'll talk about mainly is action potential, and action potential is all or nothing. Okay, now, these are the stages that we talk about when we talk about a neural impulse. And I have I have two other videos that go to this that I'm going to refer you guys to. So there's receptive, there's an initial segment, there's a conductive segment, and there's a transmissive segment. Okay, so receptive is when the neuron before comes and it releases its neurotransmitter. Okay, the neurotransmitter is going to bind to receptors somewhere on these dendrites or this cell body. Okay, this initial segment is where we're going to determine whether we fire or we don't fire. So basically what's happening is these neurotransmitters that are coming from here, okay, they could be positive, excitatory, or they could be negative and inhibitory. So they're going to bind and they're going to cause a change in the electrical potential to occur. If we sum up the change, okay, that's when we're going to determine whether we go forward or we don't go forward. So if we have enough positives, we can make the neuron fire. Okay, conductive segment's how it's going to propagate, how the message is going to get sent. And then transmissive segment is how we get to the next stage. Okay, so now I have a link from the book. Okay, and here's another link. And then this explains saltatory conduction, which we'll come back to. Okay, and then that's the end. Now what I'm going to do, I have two YouTube videos that explain all of this. They're maybe like 20 minutes long, 15 to 20 minutes long, and it explains everything in this last step. Okay, so I'll refer you to those, and then this is the rest of the chapter. Okay, now neurotransmitters in general. So when we talk about the chemical structure of a neurotransmitter, so a neurotransmitter in general is going to be made by a presynaptic neuron. They get released when that is stimulated. They're going to bind to the postsynaptic cell and they're going to change something in the postsynaptic cell. Okay, so here's my neurons, right? Okay, so this is my presynaptic and this is my postsynaptic, right? Here's the synapse. This is the synapse in here. Right, so this is the synapse. Boop, 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 boop. Okay, now this is going to release neurotransmitters, right? They're going to come in and they're going to bind. They're going to be made up here, okay, and they're going to cause a change to occur down here. That's pretty straightforward. Okay, now there's 4 types of neurotransmitters. Okay, so there are acetylcholine. Okay, there are amino acids. There are these things called biogenic amines, and then there are neuropeptides. So four main types. Okay, some of you probably maybe heard of, because you probably heard of serotonin, you probably heard of histamine, epinephrine, right? Dopamine, acetylcholine, maybe not GABA, maybe. Okay, so four main types. Okay, and the structures, if you look at them, are pretty simple. So acetylcholine is lots of times it's abbreviated ACH. Okay, neuropeptides, these are little proteins. Okay, now there are three kinds of synapses that have different ways of acting. Okay, so there are excitatory cholinergic synapse, there's inhibitory GABAergic synapse, and excitatory adrenergic synapses. This is not. Okay, so there are mechanisms to turn off simulation. Okay, so, I don't have very much about these. I thought I had more in here about the neurotransmitters. I honestly don't spend a lot of time making you memorize these. I just want you to know the four main types. They do become important if you're talking about drugs. So like GABA, different receptors and things like that, especially like mental health drugs or pain drugs. I just kind of introduce them here, so I think that's fine. Okay, now, if you need to stop a neural signal, like how do you stop it? So there are three ways, and this slide is kind of messed up. So one is the neurotransmitter could just diffuse away from the synapse, and then astrocytes kind of absorb it. Another thing is the synaptic knob could take them up, okay, and they break them down. And one example of an enzyme that breaks them down is monoamine oxidase. So if you see any like seizure drugs or antidepressants, sometimes they have MAOIs, right? So they have monoamine oxidase inhibitors in them. So basically what happens, okay, so if here's the synaptic knob and here's the next neuron, okay, and we have these neurotransmitters being released in here. So what's going to happen to them is what I'm trying to tell you. Okay, so what's going to happen to them? They can't just stay in there. If they stay in there, they would make this continue to fire. And you don't want your neurons firing all the time. You would be twitchy, right? Things would not be good if your neurons were firing over and over and over again. So basically, three different things could happen. Okay, so the neurotransmitters could just diffuse away. Okay, and they get reabsorbed by another cell. Okay, this synaptic knob reabsorbs them and can break them down. Okay, there's another enzyme called acetylcholine esterase, and it breaks down this acetylcholine in the synaptic cleft. I should have had more in here about acetylcholine. We'll get to it. Okay, so this is the same slide. This is a repeat. I don't know what I did. And that's the same slide again. What happened? Oh no. Okay. So basically that's the end of it, right? So these are three things that can happen to stop a neuron from firing. Now, let me look in this chapter real fast. If you made it to the end. Okay, we're gonna, well, there's more stuff in here, but I have a lot of this in the other PowerPoint, more detailed information about the action potential. And I think that's good. Okay. Oops, sorry guys.